Hello everyone, welcome to my channel Biochemistry Concepts. This video is about metabolism of VLDL, that is very low density lipoproteins, and LDL, low density lipoproteins. Coming to the basic function of VLDL, like chylomicrons, this VLDL also transports triacylglycerols, but it transports from liver to extrahepatic tissues. Chylomicrons, they transport from intestine to extrahepatic tissues. So, the major lipid carried by chylomicrons and VLDL is triacylglycerol, but the source is different. To understand the function of VLDL in a simple way, so we can compare the city bus services with the function of VLDL. So, when you see the city buses, they transport people within a city. They won't move out of the cities. In the same way, VLDL transports lipids which are synthesized within our body. They are not involved in the transport of dietary or exogenous lipids. So, the basic function of VLDL is they are involved in the transport of endogenous lipids. So, the major lipid carried by VLDL is triacylglycerol that is synthesized within the liver. So, as we discussed under function, VLDL is involved in the transport of triacylglycerols from liver to the extrahepatic tissues. So, this VLDL is synthesized in the liver, and that synthesis requires ApoB100 protein, which is synthesized in the rough endoplasmic reticulum, and also triacylglycerols, cholesterol, and phospholipids, which are synthesized in the smooth endoplasmic reticulum. So, ApoB100, which is synthesized in the rough endoplasmic reticulum, is incorporated into particles with triacylglycerol, cholesterol, and phospholipids in the smooth endoplasmic reticulum. After addition of carbohydrate residues in the Golgi apparatus, they are released from the cell by reverse pinocytosis. So, VLDL are secreted into the space of disc and then into the hepatic sinusoids through fenestrae in the endothelial lining. Now, this VLDL which is secreted is called as the nascent VLDL because this is functionally incomplete. This is not a completely active VLDL. So, in the second reaction, that is conversion of nascent VLDL to mature VLDL, which takes place in the circulation. So, this transformation of inactive to active form of VLDL requires the addition of two apoproteins, that is ApoC2 and ApoE. These two are supplied by HDL. Now, these mature VLDL enters into the capillaries of extrahepatic tissues. So, these capillaries, they contain an enzyme called as lipoprotein lipase, which is anchored to endothelium by heparan sulfate. So, this lipoprotein lipase hydrolyzes the triacylglycerols of VLDL, releasing the free fatty acids and glycerol. The triacylglycerols are not directly converted into free fatty acid and glycerol. So, there is a progressive hydrolysis of triacylglycerols by lipoprotein lipase. So, that converts them into free fatty acids and glycerol. So, initially this lipoprotein lipase acts on triacylglycerol and converts that into diacylglycerol, releasing one fatty acid. Again, lipoprotein lipase acts on diacylglycerol and releases one more fatty acid and forms monoacylglycerol. And this monoacylglycerol in the next reaction again acted upon by lipoprotein lipase to form glycerol plus fatty acids. So, in this way, 3 fatty acids and 1 glycerol are released by the action of lipoprotein lipase on triacylglycerol. So, this lipoprotein lipase is activated by ApoC2 and phospholipids, whereas ApoA2 and ApoC3, they act as inhibitors of lipoprotein lipase. Most of the free fatty acids released, they are transported into the tissues, whereas some released free fatty acids, they return to the circulation attached to albumin. So, in the third reaction, that is reaction with lipoprotein lipase, results in the loss of 70 to 90% of triacylglycerols, causing decrease in the size and increase in the density of VLDL. So, after the degradation of triacylglycerols of VLDL by lipoprotein lipase, it is converted into VLDL remnant or IDL, that is intermediate density lipoprotein. IDL can also be taken up by cells through receptor mediated endocytosis that uses ApoE as the ligand. But most of the IDL is transformed into LDL, that is low density lipoprotein. During this transformation of IDL to LDL, ApoE and ApoC2 are returned to the HDL. Now, LDL contains only one apoprotein, that is ApoB100. So, when we compare LDL and VLDL, LDL contains much less triacylglycerol than VLDL, and LDLs have a higher concentration of cholesterol and cholesterol esters than VLDL. The primary function of LDL is to provide cholesterol to the peripheral tissues or return it to the liver. Approximately 30% of LDL is degraded by the extrahepatic tissues and 70% by the liver. The receptors which recognize the LDL, they are located in specialized regions called as clathrin coated pits. So when ApoB100 binds to ApoB100 receptors, the LDL receptor complex is formed. Now this LDL receptor complex is internalized by endocytosis. So a vesicle containing LDL loses its clathrin coat and fuses with other similar vesicles, which forms a larger vesicle called as endosomes. These endosome vesicles, they fuse with lysosomes. The LDL particle along with apoproteins and cholesterol esters are hydrolyzed by lysosomal hydrolases, forming amino acids and free cholesterol. So the free cholesterol is either incorporated into plasma membrane or esterified to the cholesterol ester and stored within the cell. Or this cholesterol is secreted into the intestine directly or after getting converted into the bile acids.